Got my coffee and my notes and life is good. In this video, we're going to talk about viruses. First, we'll talk about the basic structure of viruses. We'll then take a look at the life cycle of viruses that infect eukaryotic cells. And then we'll have a small conversation about the role of polymerases in the replication of viral genomes. Now, a quick note on terminology here. I am referring to this image as a viral particle. Viral particles are the actual physical structure of a virus. We think of the virus as actually what's happening inside of a host cell. This particular image here, shown in light blue and a darker blue and orange, is the viral particle for poliovirus. This is what we would classify as an icosahedral virus. And typically when we draw these icosahedral viruses, we draw them as hexagons that are shown on the bottom here. So let's take a look at a couple of the different styles of viral particles that we see. As I mentioned, these two are icosahedral, which shown in black is what's known as the capsid. The capsid is made of protein, and it essentially is kind of like a nucleus. It protects the genetic material for the viral particle. The capsid on the right-hand side is coated by what we call an envelope. The envelope is made of a phospholipid bilayer that is produced from the host cells from which the virus comes. The virus can gain this envelope as it is budding out of the host cell or exiting out of the host cell. All viruses are going to have what's known as spike proteins. These are proteins that bind to receptors on the surface of host cells and are going to be required for attachment and potentially also entry and uncoding of the virus, which we'll talk about in the next couple of slides. Inside of the virus, or the viral particle, we will find the genome, which is being shown here in this hot pink color. Genomes can be either made up of DNA or RNA. They can be segmented, as is shown on the one on the right, or they can be monopartite. In other words, they can be a single double-stranded DNA or single-stranded RNA particle, either in a linear form, as is shown on the left, or in the circular form, kind of like a plasmid. Finally, there are some viruses that have what we call a helical capsid shape. These also can be enveloped, but what we find is that the genome for these viruses are typically RNA genomes, and they are oftentimes located on the inside of the protein that make up the helical capsid. Let's now go ahead and talk about the viral life cycle. Typically, we consider there to be roughly 6 steps to this life cycle. The first step is what we call attachment. And in this step here, what we find are that the spikes on the viral particle are going to bind to receptors on the host cell. Next, we have penetration, in which the viral particle now enters the host cell. This can occur by endocytosis or by fusion. Largely, this depends on what virus we're talking about. The third step is known as uncoding. During this step, the viral particle will release proteins and genome into the cytoplasm. The fourth step is known as biosynthesis. In this step, we find multiple things that are occurring that are related to DNA, RNA, and protein. First, the viral genome is going to be replicated. This could occur in the nucleus, the cytoplasm, or some other organelle. For example, some viruses actually replicate in the endoplasmic reticulum. mRNA will be synthesized, and then finally protein that produce new viral proteins are going to be synthesized as well. After the genomes are synthesized and the proteins are synthesized, we find the viruses beginning to assemble. In this step, the viral proteins are going to basically interact with each other and bind with each other, or self-assemble to form new viral particles. The viral genome is going to be packaged in capsids, and then the viral particles mature to a point where they are infectious again for the next round of a life cycle. In our final step, we have release. And in this step, the viral particles exit the host cell by processes such as budding or by lysing the host cell. In total, what we find in these 6 steps are the general process for any virus. However, it's important to point out that the life cycles are not as clear-cut as are laid out in these 6 steps. For example, penetration and uncoding may occur at the same time. Furthermore, assembly and release may occur in the order that is shown here, but maturation of viral particles may not occur until after release. And so though this is a great framework for any virus, we need to know that there are going to be special cases or instances where this process may deviate slightly. It's also important to note that this is what we call the lytic viral life cycle, where more viral particles are being created. Some viruses, like the double-stranded DNA virus family, the herpes viruses, can go into a viral latency. After uncoding, their genome will enter the nucleus, where it can then remain dormant. As host cells replicate, the viral genome is replicated alongside them, and without viral mRNA production, there will be no new viral particles formed. In fact, what we find is that in these viruses that undergo latency, they are reactivated into the lytic life cycle during periods of stress. One of the fascinating things about viruses is the variety of genomes that they may have. In living organisms, such as prokaryotes and eukaryotes, we find that the only genetic material that is inheritable from one generation to the next is DNA. But viruses can have as their genome DNA like us, but they can also have RNA as a genome. And some of them even take RNA and convert it into DNA during their life cycle. And so for each of these viruses then, they're going to require a different polymerase to make copies of those genomes. For example, viruses that have double-stranded DNA or single-stranded DNA genomes are going to require what we call a DNA-dependent DNA polymerase. The first part of this name, DNA-dependent, tells us what the template is for this polymerase. The second part, DNA polymerase, tells us what will be made. So for example, in these 2 viral cases here, double-stranded DNA and single-stranded DNA, they use DNA as a template to make more DNA. And in fact, the eukaryotic and the prokaryotic DNA polymerases are DNA-dependent DNA polymerases. In some cases, these double-stranded and single-stranded DNA viruses may use host polymerases in order to perform replication and transcription. Now, I want you to take a look at the mutation rate here. For viruses that encode their own DNA-dependent DNA polymerase, those polymerases make a mistake maybe once every 10 million bases that are replicated, or 1 in every 10 to the 7th bases. These polymerases are incredibly high fidelity. Compare this to RNA viruses that encode RNA-dependent RNA polymerases. They use RNA as a template to make more RNA. The mutation rate of these polymerases is somewhere on the order of 1 in 10 to the 4th to 10 to the 5th. In other words, for every 10,000 to 100,000 bases, these viruses are making at least one mistake in replication. Retroviruses, like HIV, are viruses that take their RNA genome and converted into DNA before integrating that DNA into the host genome. They require an enzyme known as reverse transcriptase, which is an RNA-dependent DNA polymerase. This reverse transcriptase enzyme is low fidelity, making one mistake in every 10,000 bases. The rate of mutation that we find here have really big implications for biology, but also for medicine. First, what this tells us, since mutations are one of the driving factors of evolution, is that RNA viruses typically evolve at a quicker rate than do DNA viruses. Secondly, this makes it more challenging to eradicate viruses that have RNA genomes. In fact, we find that because their genomes are more unstable, the vaccines that we may have used in the past may not necessarily work now to treat these viral pathogens. So let's finish off with the comparison in the biosynthesis of DNA and RNA viruses. DNA viruses undergo replication to produce more DNA with help from a DNA-dependent DNA polymerase. This polymerase may be encoded for by the virus, or it may be encoded for by the host. It just depends on the particular virus. Next, that DNA can be transcribed into RNA using a DNA-dependent RNA polymerase. And finally, that mRNA can be translated in the cytoplasm to a protein. And this requires the host's ribosome in order for translation to occur. In RNA viruses, what we find is that RNA will be replicated to produce more RNA. And that involves a viral RNA-dependent RNA polymerase. That same polymerase can be used to transcribe the RNA genome into mRNA, and that mRNA is then translated in the cytoplasm to protein, again using the host's ribosome. As we discuss different viruses in the future, this will be a key step that we will focus on to distinguish different viruses from one another.